Hi everyone and welcome to BU Pharma Insights. Here I am starting a series of 62 videos on the topic Advanced Pharmaceutical Dissolution Science and Application. First 12 videos are related to core fundamental understanding of pharmaceutical dissolution science. In this video, we will discuss what is dissolution. Actually, dissolution tells us how fast and how much of the drug is released from its dosage form into liquid medium. We perform dissolution testing to simulate what happens in the stomach or intestine using carefully selected media, temperature, and agitation that mimic in vivo conditions. Dissolution is only about drug release, not absorption. A drug must first dissolve before it can absorb. Absorption happens after dissolution, not during it. So, dissolution is essentially the step just before drug enters the bloodstream. In simple words, we are checking how quickly and how completely a drug comes out the tablet or capsule before body can absorb it. Now, why do we need dissolution? Whenever you swallow a tablet, a capsule, the first thing it must do is release the drug. Dissolution testing tells us how fast and how completely the drug comes out of the dosage form inside the body. It helps ensure the product is safe, effective, and consistent, while also allowing us to identify formulation issues early, support regulatory submissions, and confirm performance during stability. In simple terms, dissolution mimics what happens in the stomach or intestine, how quickly the drug dissolves and becomes available for absorption. Now, difference between dissolution and disintegration. Many beginners confuse disintegration with dissolution. Disintegration is when tablet breaks into smaller pieces, while dissolution is when the drug molecules actually dissolve into the medium. A tablet may disintegrate quickly but still dissolve slowly if drug is poorly soluble. So these two tests measure completely different things. Now, factors that influence dissolution. The rate and extent of dissolution depend on three categories of factors. First, drug properties such as solubility, particle size, and polymorphism. Second, formulation factors like excipient, tablet hardness, coating, and manufacturing process. And third, testing conditions such as pH of the medium, rotation speed, and type of apparatus used. These factors decide how a drug dissolves and why different tablets behave differently. Now, dissolution apparatus in brief. In the laboratory, we use different USP apparatus to perform dissolution. Apparatus 1, called the basket, is mainly used for capsules. Apparatus 2, the paddle, is widely used for the tablets. Apparatus 4, known as flow-through cell, is used for complex dosage form. Other apparatus like 5, 6, 7 are used for modified applications. For most routine products, apparatus 1 and 2 are sufficient. So, what does a typical dissolution test measure? A typical dissolution test gives us the percentage of drug release at different time points like 5, 10, 20, or 30 minutes. We often use a criteria such as Q equal to 80% in 30 minutes, which means at least 80% of drug must dissolve by 30-minute mark. The result is usually plotted as a dissolution profile, showing drug release over time. Now we'll discuss a bit on sink conditions. A very important concept in dissolution testing is sink condition. This means the dissolution medium must be able to dissolve at least 3 times of the amount of the drug present in the dosage form. If medium becomes saturated too quickly, Release slow down and results become misleading. Now we will take a real life example. Let's take a simple example, paracetamol. When you take the tablet, it first breaks down, then drug dissolve in the stomach fluid, and finally it gets absorbed. Dissolution testing in the lab helps us to check whether every batch of paracetamol tablets releases the drug at right speed and in the right amount. This ensures consistent quality and predictable therapeutic effect. So, after having a preliminary discussion, we should know why dissolution is critical in pharmaceutical industry. Dissolution is one of the most critical tests in the pharmaceutical industry. It ensures product performance, safety, and batch consistency. It also helps us to identify formulation or process issues early. Regulatory agencies like FDA, EMA, and WHO rely heavily on dissolution data for product approvals and life cycle management. Next is application across product life cycle. Dissolution is not just a QC test, it is used throughout the entire product life cycle, from formulation development, scale up, and technology transfer to routine QC testing and stability studies. It also supports change control, post-approval variations, and even bioequivalence predictions. So, dissolution plays a role in almost every stage of drug journey. Now I'm summarizing this presentation here. Dissolution tells us how quickly and how much drug is released from a dosage form. It is influenced by drug properties, formulation factors, and testing conditions. It is essential for the product development, quality control, and regulatory approvals. Understanding dissolution is the first step toward mastering pharmaceutical testing. Thank you for watching. Please subscribe us for getting update on such industry application oriented videos, and stay tuned because in this 62 video series on dissolution science we will cover dissolution method development, validation, troubleshooting, and many real industry examples. In the next video we will focus on biopharmaceutics basics, why dissolution testing matters. If you have any question, post them in comment section. I will do my best to respond. See you in next video. Thanks.